Let's start with today's featured paper. Ameliorating calcium homeostasis improves longevity and health span in progeroid and naturally aged mice. By Weifang Xiang and Yu Zhang from Key Laboratory of Molecular Epigenetics of Ministry of Education, MOE, Northeast Normal University, Changchun, China. This paper was selected because it uncovers a novel and fundamental molecular mechanism linking disrupted calcium homeostasis to aging and progeria, demonstrating that targeting this pathway can significantly improve longevity and health span in mice. Here's a full breakdown. Next up, ATOX1 deficiency induces memory impairment via promoting. Today, we're diving into a fascinating study that explores the critical role of calcium regulation in the aging process. Many age-related conditions are associated with issues in how our cells manage calcium. This research aimed to uncover the specific mechanisms by which disrupted calcium homeostasis contributes to both natural aging and premature aging syndromes like progeria, and importantly, whether we can intervene to improve health and longevity. The researchers employed a multifaceted approach, utilizing both progeroid and naturally aging mouse models, alongside human cells from HGPS patients and healthy aged individuals. They meticulously investigated cellular calcium dynamics, protein interactions, and key signaling pathways like C-gas sting and F-kappa B. Their methods allowed them to pinpoint how calcium dysregulation leads to a cascade of events, including DNA damage and inflammation, ultimately contributing to aging phenotypes. They also tested a specific compound, myanserin, to see if it could reverse these effects. The findings were quite striking. They observed that disrupted calcium homeostasis directly leads to the accumulation of a protein called S100A6, which then promotes both Hutchinson-Gilford progeria syndrome in natural aging. Crucially, the drug myanserin was shown to effectively attenuate cellular senescence in human cells and, even more impressively, significantly extended the lifespan and improved various aging phenotypes in both progeroid and naturally aging mice. This was achieved by myanserin's ability to lower calcium concentrations by targeting specific serotonin receptors. This research offers profound insights into the aging process, identifying calcium dysregulation as a central driver. The discovery of the S100A6, PRP1C gas, sting pathway provides a new, actionable target for developing therapies. The most exciting aspect is the identification of myanserin, an existing drug, as a potential intervention that significantly extended lifespan in animal models. While these findings are highly promising, further studies are essential to translate these results into safe and effective treatments for human aging and progeroid syndromes. That was our featured study. Today, PubMed indexed 862 new open access papers across 338 journals. We've selected 10 trending research areas and 12 studies for today's video. Let's look at the trending topics before we continue. Today's research is buzzing with health breakthroughs, from global interventions to AI's role in clinical medicine, alongside notable progress in mental health and environmental engineering. Now, let's see which papers stood out. Forget what you thought you knew about evolution. Horizontal gene transfer is far more common in eukaryotes than previously believed. And imagine your smartwatch predicting early hypertension, offering a new path to preventative health. Now let's explore each of today's trends in detail, starting with number 1. Trend 1 of 10, Global Health Interventions and Outcomes. Research evaluating health interventions and their outcomes across diverse global populations and settings. This cluster covers 87 papers. Representative work includes effectiveness of breastfeeding educational interventions during pregnancy on breastfeeding practices in rural central Ethiopia. Cluster randomized controlled trial, patient-centered care, and non-operative versus operative management of uncomplicated acute appendicitis, a randomized controlled study in Sierra Leone. Trend 2 of 10, Crop improvement and environmental engineering. This trend combines genetic and engineering approaches to enhance agricultural productivity and manage environmental challenges. This cluster covers 59 papers. Representative work includes comparative nutritional analysis of newly developed interspecific citrus hybrids, multivariate insights for crop improvement, integrating gene expression and morphological traits for drought stress adaptation in maize hybrids, and zoonotic malaria in Southeast Asia's changing landscapes. Vector complexity, challenges, and future strategies. Trend 3 of 10, Cancer mechanisms and radiosensitivity. Investigating cancer development mechanisms and factors influencing its response to radiation therapy. This cluster covers 93 papers. Representative work includes exploring the high-risk factors of gastric inflammatory cancer transformation based on inflammatory immune response, Construction of RHPI complicated with CAG risk prediction model. Calcium overload induces reactive oxygen species to enhance glioblastoma radiosensitivity, and ionomycin enhances radiosensitivity in breast cancer through reactive oxygen species generation and mitochondrial permeability transition, pore opening. Trend 4 of 10, Neuroscience and Neurological Disorders. This trend explores neuroscientific mechanisms, biomarkers, and diagnostic approaches for various neurological disorders and brain functions. This cluster covers 73 papers. Representative work includes infections as part of the multiple sclerosis prodrome, a systematic review, transcriptional correlates of structure-function coupling plasticity in trigeminal neuralgia, unveiling the synaptic and metabolic associations, and brain neuromarkers predict self and other related mentalizing across adult, clinical, and developmental samples. Trend 5 of 10, Applied Biology and Medical Research. This trend encompasses research applying biological principles and methods to solve practical problems in medicine, agriculture, and biotechnology. This cluster covers 147 papers. Representative work includes establishment and validation of reference intervals for the neutrophil CD64 index in a healthy adult population from Guangxi, China, from lab to field, accelerated cultivar release via integrated speed breeding framework in wheat, Triticum estivamel, and enriched trial of preservative-free lubricants in dry eye disease. Trend 6 of 10, Public Health and Lifestyle Factors. Investigating how lifestyle factors like diet, activity, and substance use influence public health outcomes and individual well-being across diverse populations. This cluster covers 55 papers. Representative work includes nutrition literacy, dietary diversity, and frailty among older adults in China, randomized controlled feasibility trial of an intervention to increase activity and reduce sedentary behavior in people with severe mental illness, Walking for Health, Worth, Study, and associations of cumulative exposure and dynamic trajectories of the combined atherogenic and frailty index with incident cardiometabolic multimorbidity, a longitudinal analysis based on the China Health and Retirement Longitudinal Study, CHARLS. Trend 7 of 10, Clinical Medicine and AI Applications. This trend explores diverse clinical medicine topics, often leveraging artificial intelligence for improved analysis, prediction, and patient care. This cluster covers 152 papers. Representative work includes age-related patterns in hysterectomy, indications and surgical route selection in a 3-year tertiary center cohort, deep learning for predicting cardiac procedure outcomes, a scoping review of recent advances, and lactobacillus-containing probiotics and clinical pregnancy in infertile women undergoing ART, a systematic review and meta-analysis. Trend 8 of 10, Computational Science and Data Engineering. This trend explores computational methods and data engineering techniques for modeling, simulation, and analysis across various scientific and engineering applications. This cluster covers 82 papers. Representative work includes a multimodal dataset of full-body kinematics and kinetics from laboratory and wearable-based systems, comparative DFT investigation of 1T and 2H phase Janus TAS monolayers as potential sensors for toxic gas molecules, and algorithm selection for lithium-ion battery ECM parameterization, a comparative study of trust region, Levenberg-Marquardt, Gauss-Newton, and BFGS for accuracy and physical consistency. Trend 9 of 10, Mental Health and Behavioral Studies. Examines mental health conditions, behavioral patterns, and their influencing factors across diverse populations and contexts, including animal models. This cluster covers 65 papers. Representative work includes depression, pain, and functional impairment across fibromyalgia, rheumatoid arthritis, and systemic lupus erythematosus, a cross-condition analysis and mediation model, developmental and cultural factors in bulimia nervosa, a comparative study of Croatian and German female patients across age cohorts, and risk of insomnia disorder in Japanese cancer patients, a matched cohort study using employer-based health insurance claims data. Trend 10 of 10, Orthopedics, Anatomy, and Imaging. Studies on biological structures, their properties, and related medical interventions, often utilizing analytical methods. This cluster covers 49 papers. Representative work includes an anatomical study on the characteristics of the shape of the foot and talofibular and calcaneofibular ligaments in relation to the different number of bundles of the anterior talofibular ligament and its relevance, effect of fluorescent adhesives on visibility and tooth enamel surface properties after debonding, a systematic review and meta-analysis, and a clinical applicable study on lower limb segmentation from CT images for total knee arthroplasty. That's the full research landscape for today. Remember, our number one paper, Ameliorating Calcium Homeostasis Improves Longevity and Healthspan, was already covered at the top. Now let's continue with papers 2 through 12, one by one. Up next, our second paper out of 12, ATOX1 deficiency induces memory impairment via promoting cuproptosis in Alzheimer's disease by Haitao Yu and Gaoshang Chai from Department of Fundamental Medicine, Wuxi School of Medicine, Jiangnan University, China, Department of Forensic Medicine, Guizhou Medical University, Guiyang, Guizhou, China. This paper was selected because it uncovers a novel mechanistic pathway involving ATOX1 deficiency and cuproptosis in Alzheimer's disease, offering a fundamental biological discovery that could lead to entirely new therapeutic strategies for this devastating neurodegenerative condition. Let's break down its key contributions. Next up, anthropometric outcomes at 12 years after fresh. Alzheimer's disease, a devastating neurodegenerative disorder, is often linked to disruptions in copper balance within the brain. We know that too much copper can worsen brain damage and memory problems. Recently, a new form of cell death called cuproptosis, driven by copper overload, has emerged, but its exact involvement in AD has been a mystery. This study aimed to shed light on this crucial connection. To unravel this mystery, our team employed a multifaceted approach. We analyzed human AD brain samples and utilized both AB/PS1 mouse models and A-beta-treated HT22 cells to mimic AD pathology. We used a range of advanced techniques, including proteomics and behavioral tests, to assess molecular changes and cognitive function. Crucially, we manipulated ATOX1 levels using genetic tools to understand its direct impact on disease progression. Up next, our third paper out of 12, anthropometric outcomes at 12 years after fresh versus frozen embryo transfer compared with natural conception by Kinsuka Kitsu and Naoki Kuji from Department of Obstetrics and Gynecology, Tokyo Medical University, 671 Nishi Shinjuku, Tokyo 160, Japan. This paper was selected because it provides crucial long-term, multi-center prospective cohort data on the anthropometric outcomes of children conceived via assisted reproductive technology, directly addressing a pressing clinical and public health concern regarding the safety and long-term effects of these widely used interventions. Let's break down its key contributions. Next up, rapid development and field evaluation. We've seen some evidence that babies conceived via frozen thawed embryo transfer, or FET, might be larger at birth. This raises an important question. Does this difference in size persist as these children grow older? Understanding the long-term anthropometric outcomes is really critical for families considering ART and for guiding clinical practice. This study aimed to shed light on this by following children up to 12 years of age. This was a multi-center prospective cohort study conducted in Japan, following singleton children born between 2008 and 2009. Researchers compared children conceived via fresh embryo transfer, frozen thawed embryo transfer, and natural conception. The primary outcomes were anthropometric measurements like weight, height, BMI, and the prevalence of overweight or obesity when the children reached 12 years of age. The study carefully adjusted for various confounding factors to ensure robust comparisons. Up next, our fourth paper out of 12, rapid development and field evaluation of a portable CRISPR-based assay for Mpox during the 2025 Sierra Leone outbreak by Nisha Gopal and Party C. Sabeti from Broad Institute of Massachusetts Institute of Technology and Harvard, Boston, Massachusetts, USA. This paper was selected because it demonstrates the rapid development and successful field evaluation of a portable CRISPR-based diagnostic during an active outbreak, addressing a critical global health need for accessible and deployable pathogen detection. Let's break down its key contributions. Next up, optimization of IS621 recombinase/bridge RNA-directed recombination. The recent Mpox outbreak in Sierra Leone underscored a critical challenge, getting rapid, accurate diagnostics to where they're needed most. Traditional lab tests can be slow and resource-intensive, leaving many communities without timely access. This paper tackles that problem head-on, exploring how cutting-edge CRISPR technology could offer a solution for rapid, decentralized testing. Researchers rapidly developed Mpox Shine, a novel CRISPR-Cas13 assay designed for portability. They then rigorously evaluated its performance in the field during the 2025 Mpox outbreak in Sierra Leone. The study compared Mpox Shine's accuracy against the gold standard qPCR using 56 clinical specimens, assessing its ability to correctly identify both positive and negative cases. Up next, our fifth paper out of 12, Optimization of IS621 recombinase/bridge RNA-directed recombination for precise insertion of large DNA fragments in human cells by Junfan Guo and Lina Wu from School of Life Science and Technology, Shanghai Tech University, Shanghai, China. This paper was selected because it introduces a novel and optimized gene editing technology that enables the precise insertion of large DNA fragments into human cells, addressing a critical limitation in current gene therapy approaches and opening new avenues for treating a wide range of genetic diseases. Let's break down its key contributions. Next up, prognostic data extraction harnessing a privacy-preserving large. Gene therapy holds immense promise, but precisely inserting large DNA fragments into specific locations in human cells has been a significant hurdle. The IS621 recombinase, a fascinating molecular tool from bacteria, offers a unique way to achieve sequence-specific DNA recombination. This study aimed to adapt and optimize this powerful system for use in human cells, addressing a critical need in gene editing. Researchers embarked on a journey to engineer the IS621 recombinase and its associated bridge RNA. They systematically developed a new system called NS621 tebRNA, specifically designed for human cells. This innovative approach allowed them to achieve precise, scarless integration of large DNA fragments, which was a major technical breakthrough. You're halfway through. Still coming up. This paper was selected because it presents a groundbreaking challenge to the long-held biological dogma that horizontal gene transfer is rare in eukaryotes, potentially redefining our understanding of eukaryotic evolution and gene function. This paper was selected because it presents a groundbreaking challenge to the long-held biological dogma that horizontal gene transfer is rare in eukaryotes, potentially redefining our understanding of eukaryotic evolution and gene function. This paper was selected because it introduces a highly innovative biomimetic nanocarrier system for mucosal vaccination, directly addressing critical barriers to effective pulmonary immune responses against respiratory pathogens, which represents a significant advancement in vaccine delivery technology. Keep watching. The second half is just as strong. Up next, our sixth paper out of 12, Prognostic Data Extraction Harnessing a Privacy-Preserving Large Language Model. A clinician-AI collaborative retrospective evaluation in head and neck oncology by Yujing Zhou and Sharon A. Anger from Medical Physics Unit, Department of Oncology, Faculty of Medicine, McGill University, Montreal, Quebec, Canada. This paper was selected because it introduces a highly accurate, privacy-preserving methodology for extracting complex prognostic variables from unstructured clinical notes using large language models, which directly addresses a critical and widespread bottleneck in clinical research and AI development. Let's break down its key contributions. Next up, horizontal gene transfer is widespread and diverse. LLMs hold immense promise for healthcare, but their deployment is often hampered by strict privacy regulations and the need for expert validation. This creates a significant challenge in leveraging these powerful tools for tasks like extracting critical information from electronic health records. Our study aimed to address this by evaluating how effectively locally deployed LLMs could extract prognostic variables from head and neck cancer reports in a privacy-preserving way. We conducted a retrospective evaluation using 360 clinical reports from 882 head and neck cancer patients. Our primary intervention involved using various locally deployed large language models, including Llama 3.3-70B, to perform zero-shot extraction of 30 prognostic variables. To establish a gold standard, 3 radiation oncologists created a majority vote reference on a subset of 50 cases. We then compared the LLM's extraction accuracy against this expert-validated reference and assessed their impact on prognostic model performance. Up next, our 7th paper out of 12. Horizontal gene transfer is widespread in diverse eukaryotes by Kuan Li and Chao-Chun Wei from School of Life Sciences and Biotechnology, Shanghai Jiaotong University, 800 Dongchuan Road, Shanghai, China. This paper was selected because it presents a groundbreaking challenge to the long-held biological dogma that horizontal gene transfer is rare in eukaryotes, potentially redefining our understanding of eukaryotic evolution and gene function. Let's break down its key contributions. Next up, prognostic data extraction harnessing a privacy-preserving large. Horizontal gene transfer, or HGT, is a fascinating process where genetic material moves between distantly related organisms. While we know it's widespread in bacteria, its role in eukaryotes, like humans or plants, has largely been considered rare and limited. This study aimed to challenge that assumption and investigate just how common HGT truly is across the eukaryotic tree of life. It's a fundamental question with big implications for how we understand evolution. To explore this, researchers conducted a comprehensive genomic analysis across 10 diverse eukaryotic species, from humans to yeast. They used advanced techniques like third-generation sequencing and targeted PCR to identify and validate horizontal gene transfer events. This robust approach allowed them to meticulously compare eukaryotic genomes against a vast database of bacterial and viral genetic material, ensuring the accuracy of their findings. Up next, our 8th paper out of 12, Prognostic Data Extraction Harnessing a Privacy-Preserving Large Language Model. A clinician-AI collaborative retrospective evaluation in head and neck oncology by Yujing Zhou and Sharon A. Anger from Medical Physics Unit, Department of Oncology, Faculty of Medicine, McGill University, Montreal, Quebec, Canada. This paper was selected because it introduces a highly accurate, privacy-preserving methodology for extracting complex prognostic variables from unstructured clinical notes using large language models, which directly addresses a critical and widespread bottleneck in clinical research and AI development. Let's break down its key contributions. Next up, horizontal gene transfer is widespread and diverse. LLMs hold immense promise for healthcare, but their deployment is often hampered by strict privacy regulations and the need for expert validation. This creates a significant challenge in leveraging these powerful tools for tasks like extracting critical information from electronic health records. Our study aimed to address this by evaluating how effectively locally deployed LLMs could extract prognostic variables from head and neck cancer reports in a privacy-preserving way. We conducted a retrospective evaluation using 360 clinical reports from 882 head and neck cancer patients. Our primary intervention involved using various locally deployed large language models, including Llama 3.3-70B, to perform zero-shot extraction of 30 prognostic variables. To establish a gold standard, 3 radiation oncologists created a majority vote reference on a subset of 50 cases. We then compared the LLM's extraction accuracy against this expert-validated reference and assessed their impact on prognostic model performance. Up next, our 9th paper out of 12. Horizontal gene transfer is widespread in diverse eukaryotes by Kuan Li and Xiaochun Wei from School of Life Sciences and Biotechnology, Shanghai Jiaotong University, 800, Dongchuan Road, Shanghai, China. This paper was selected because it presents a groundbreaking challenge to the long-held biological dogma that horizontal gene transfer is rare in eukaryotes, potentially redefining our understanding of eukaryotic evolution and gene function. Let's break down its key contributions. Next up, innovative mucosal nanocarrier systems for enhanced immune. Horizontal gene transfer, or HGT, is a fascinating process where genetic material moves between distantly related organisms. While we know it's widespread in bacteria, its role in eukaryotes, like humans or plants, has largely been considered rare and limited. This study aimed to challenge that assumption and investigate just how common HGT truly is across the eukaryotic tree of life. It's a fundamental question with big implications for how we understand evolution. To explore this, researchers conducted a comprehensive genomic analysis across 10 diverse eukaryotic species, from humans to yeast. They used advanced techniques like third-generation sequencing and targeted PCR to identify and validate horizontal gene transfer events. This robust approach allowed them to meticulously compare eukaryotic genomes against a vast database of bacterial and viral genetic material, ensuring the accuracy of their findings. Up next, our 10th paper out of 12, Innovative Mucosal Nanocarrier Systems for Enhanced Immune Response Against Respiratory Pathogens by Zhenjin Zhang and Zhixin Feng from Institute of Veterinary Medicine, Jiangsu Academy of Agricultural Sciences, Nanjing, China. Veterinary Biological Products, Taizhou, Huatai Technology Innovation Center, Taizhou, China. This paper was selected because it introduces a highly innovative biomimetic nanocarrier system for mucosal vaccination, directly addressing critical barriers to effective pulmonary immune responses against respiratory pathogens, which represents a significant advancement in vaccine delivery technology. Let's break down its key contributions. Next up, engineering a PD-L1 sensing synthetic receptor for programmable. Pulmonary vaccination holds great promise, but it faces significant hurdles. Our lungs have natural defenses, like the pulmonary surfactant barrier, that make it difficult for vaccines to reach their targets, especially the alveolar macrophages. This inefficiency limits how well our immune system can respond. This research set out to tackle these challenges head-on, aiming to create a new nanovaccine platform that could bypass these barriers and deliver a powerful immune punch. To address these challenges, researchers developed biomimetic alveolar macrophage membrane vesicles, or AMVs. These AMVs were engineered with an antigen capture and cytosolic delivery system, or ACCDS, which included components like an engineered surfactant protein domain and a pH-responsive listeriolysin O module. This innovative design aimed to improve delivery, facilitate intracellular escape, and activate innate immunity. The platform was then tested in both in vitro models and in vivo animal studies, including prime boost immunization protocols. Up next, our number 11 paper out of 12, Engineering a PD-L1 Sensing Synthetic Receptor for Programmable Macrophage Response by Ilaria De Martino and Valia Siciliano from Istituto Italiano di Tecnologia IT, Largo Barsani Matucci, Naples, Italy. This paper was selected because it introduces a highly novel synthetic biology approach to engineer immune cells with programmable responses, representing a significant advancement in targeted cellular therapies for complex diseases. Let's break down its key contributions. Next up, Smartwatch-derived exercise metrics as predictors of early. Our cells are constantly making complex decisions based on signals from their environment. Synthetic biology gives us the power to reprogram these decisions by designing new receptors. This study focuses on a key immune regulator called PD-L1, which is often targeted in cancer therapies. The goal was to turn PD-L1 from just a target into a programmable input for cellular responses. The researchers designed a novel synthetic notch-based receptor, or SNP, to specifically detect PD-L1. They tested this system in monocytic-like cells, specifically THP-1-derived macrophages, and also used SKOV3 ovarian cancer cells to evaluate its impact. The SNP was engineered to trigger programmable outputs, such as expressing a fluorescent reporter or a specific effector molecule, CV1-FC, upon sensing PD-L1. All these experiments were conducted in a controlled laboratory setting, in vitro. Up next, our number 12 paper out of 12, Smartwatch-derived exercise metrics as predictors of early hypertension. A prospective observational study by Esma Selva Atesh Sihun and Gökhan Sihun from Department of Cardiology, Erzurum City Hospital, Erzurum, Turkey. This paper was selected because it introduces a highly scalable and non-invasive method using consumer smartwatches to predict early hypertension, offering a novel and broadly accessible approach to preventative cardiovascular health. Let's break down its key contributions. Hypertension is a major risk factor for cardiovascular disease, and catching it early is crucial for prevention. This study explored whether everyday smartwatches, which track various physiological and activity metrics, could help us identify individuals at higher risk for developing hypertension before it becomes a clinical problem. It's about finding new, accessible ways to predict health risks. Researchers conducted a 12-month prospective observational study, following 230 normotensive adults between 30 and 60 years old. They used consumer smartwatches to collect data on heart rate variability, resting heart rate, and physical activity. The main goal was to see if these smartwatch metrics could predict the development of hypertension, defined by European guidelines, over the study period. That's all for today's PubMed Daily. If you found this helpful, please like, leave a comment, and subscribe for daily paper summaries. See you tomorrow.